Stop moving the mouse. Yep. Hide video panel as well.
Okay, hello everyone. Welcome back to our second session of the morning. Now we have James Fairbanks, who's here to talk about compositional account of motifs, mechanisms, dynamics in biochemical regulatory networks.
Great, thanks. So this is joint work with a group of people, Rebecca, Amit, Pablo, Evan, and Brandon. And we are, so we did this, started this project at the AMS math research community last year. I see several people here who also attended. We had a great time at this place called Beaver Hollow, New York, which is like this idyllic place to hang out and do math for a week. And they served the official dessert of applied category theory, lemon meringue pie. And it was just a coincidence. I thought it was the first day, so I was like, we're gonna have lemon meringue pie every day. It's gonna be great. They got our dessert figured out, but it was just random. And then, so I really want to thank all the organizers of the AMS MRC, which enabled this project for us to get together and collaborate. And if anybody wants to organize an AMS MRC for category theory again in 2025, let me know, and we can write a proposal for that. It was really fun. So this work fits into a broader project that I'm working on in my research group, which includes the algebraic Julia team, of understanding scientific computing technology and how at the one hand we think about arbitrary code, and at the other hand we think of computer algebra and symbolic math. And so like all of this work I'm going to talk about today fits into this, as it's kind of a computer algebra of modeling frameworks for biochemical modeling. And if you came to the algebraic Julia tutorial yesterday, you would have seen a lot of kind of what we're about and our broader vision. We have kind of, on the one hand, all possible modeling frameworks to serve scientists, and on the other hand, all possible modeling tasks that scientists want to do. And normally this is like a n squared thing. For every modeling framework, you have to implement all the tasks, right? So there's like a quadratic amount of work. And we think that applied category theory allows you to factor through a small set of reusable primitives. And then we implement those reusable primitives in software in algebraic Julia to enable like better scientific software. And it's really about this talk is about what are the reusable primitives that we were able to use in this project. Kind of more, none of you are mathematical biologists, so what I have to say about regnets is like not particularly important to you. I think what's important for us in this room is like what reusable pieces of applied category theory were successful in tackling this problem, because they'll be successful in every problem. Well, several problems. So like for context, what are the biologists doing? They're studying regulation in complex systems. So this is a figure from a paper a friend of mine, Adam McLean, who studies like how do tumors grow inside people and how do they interact with their host environment. And tumors do this really cool thing. If you're a tumor or like you're pro-cancer, they do this really cool thing where the tumor cell recruits these things called MDSC cells, and MDSC cells like shut down the immune system. So the tumor does two things. It both like causes an immune response, which is mediated by these two types of cells, NK cells and CTL cells. So it like causes, the tumor is there, it's bad, so the immune system comes in to try and kill it. But the tumor cell also recruits MDSC cells. And MDSC cells, I have no idea what that stands for, but they are pro-tumor populations. So they come in and they shut down the immune response. So these little arrows are like the arrows with the arrowheads. What's that? It does stand for myeloid-derived suppressor cells. Thank you. So the suppressor cells suppress, and they come in, and so the tumor cell like upregulates the production of MDSCs, and it upregulates the production of these anti-tumor populations. And the anti-tumor populations, because they're anti-tumor, like downregulate the tumor cells. They try to come in and destroy the tumor cells. But the MDSC cells downregulate the anti-tumor cells. So they're pro-tumor because anti-tumor is pro-tumor. And that's going to be important. So then like they make this like qualitative model of what is causing what, either up or down. That's all they can really measure is like, does it cause more of something or does it cause less of something? And then they can like build ODE models that model the dynamics of how many of each cell are in the body, right? So you have as a function of time, like the tumor is growing, it causes MDSCs to grow, the MDSCs shut down the other cells. NK stands for natural killer, which is probably the most metal thing in immunotherapy. So yeah, so they're growing and then they're causing the other things to go down. So we looked at this problem and we're like, how can applied category theory help? And we need to find biological concepts that we're gonna model with applied category theory. So there's the data of the regulatory network. That's like the different types of cells and what they upregulate and downregulate. And we're gonna use an attributed C-set for sign graphs. Then what are the regulation interactions? We're gonna model that with sign categories. We need a qualitative interpretation of these graphs. And so our graphs are gonna take functorial semantics in categories with like the path functor. Motifs are these like repeated little pieces of interaction that come up over and over again. And they're gonna be representable functors. Like they're the things that are represented by representable functors. Mechanistic explanations, we're gonna kind of think of this as an inverse problem or a lifting problem. And quantitative analysis, we're gonna take functorial semantics this time in parameterized dynamical systems. And then complex systems are like kind of complex in the sense of simplicial complex. It's like a complex of systems. So it's a bunch of little systems that are arranged into a bigger system. And we have a tool for that, structured cospans. Yeah, so I think the things on the right, you should all recognize from other talks at this conference. Like, we're not inventing any of these things. This like paradynam invented by applying a general construction and looking at the output. But yeah, so these are all like technologies that ACT has produced that are now ready to be applied to the biological problem. So just a little review of C-sets. You have a small, like finitely presented category that you're gonna think of as a schema, and you're gonna take functors into set, and you're gonna think of those as like databases over that schema. So here's the schema for graphs, and an example of a graph. Petri nets, which John probably talked about in the last talk. So you have like reaction networks, where we have states and transitions, and some of these things can be quantitative, that's the attributed part, where you can have kind of numeric data in your Petri net. So we worked with two different types of adjets, signed graphs, where we think of having vertices, edges, and every edge has a sign, and there's an involution on whatever the signs are, so there's like a negative sign. For every sign, there's an opposite sign. So if this A is like Z mod 2, then you get like signed graphs are just graphs where the edges have signs like we're going to draw them with arrowheads if they are upregulating, and the maps to symbol, the flat arrowhead, if it's downregulating. And then this is a slice category. This is equivalent to a slice category. Signed Petri nets are just like Petri nets, except they also have this set of links, and the links have signs. And so we're going to draw them like this, where we like annotate the sign. The dashed arrows are the links, and they're going to be annotated with their sign. So when we go to interpret a regulatory network, we just modeled them as C-sets. So what are we going to do for the morphisms? Well, the data is a functor, and if I have two functors, how do I compare them? I use a natural transformation. So that will give me a sign-preserving graph homomorphism, which is cool. All of those are useful, but there are other morphisms that we want that are not captured by that, which is we want to think about the logical structure of the path. If A upregulates B and B upregulates C, well then A upregulates C by the transitive property of upregulating. But if A upregulates B and B downregulates C, that's A downregulating C. Because I am creating more of a thing that suppresses you. So I suppress you by transitivity. So in order to model that, we pass two signed categories, which are categories where all the morphisms have signs, and the signs multiply, negative times negative is positive. So we take a slice category of graph and a slice category of cat over this, basically the same object, and then we can think of you generalize the path functor to the signed path functor, and then you take the Kleisli category of that. So our objects are signed graphs, but our morphisms are signed functors between their corresponding path graphs. And that lets you talk about things like, my morphisms are now allowed to send an edge to a composite of edges that have the right sign. That's super important. So now our motifs, this is where we get motifs, they can be explained, they're just figures of a shape. What's a motif? It's like a shape. So you just take Homs in this Kleisli category, you have to look at the monic ones, because you don't want to merge chemical species. So you want to be able to take an arrow to a path, but you don't want to be able to take two types of cell to the same type of cell. So you want monic arrows in this Kleisli category, and these are the little shapes. So positive autoregulation, you upregulate yourself. Negative, you downregulate yourself. A coherent feed-forward loop, you have two ways of upregulating something else. An incoherent feed-forward, you both upregulate and downregulate something. Feedback, you upregulate something that upregulates you. Negative feedback, you upregulate something that downregulates you. Double negative feedback, you downregulate something that downregulates you. Right? And so these are kind of all the graphs with only two edges, or one or two edges, right? You can kind of explore all of them, and they're all meaningful. And because we are dealing with representable functors, if you have a morphism, so you have a motif with two things, and you have another motif with three things, you can think of that as a refinement of this motif. So there's a whole, ontology is not the right word, taxonomy. There's a whole taxonomy of these motifs that are what are all of the ways you can implement an incoherent feed-forward loop? So there's incoherent feed-forward of type A, incoherent feed-forward of type B, incoherent feed-forward of type C. And they build these huge taxonomies that are all just saying does this pattern occur in your network? And now we can talk about a refinement ordering. If you want to think if there exists a morphism from S to X, then you say it's a refinement. Yeah, go ahead.
You said they built.
They built the biologists.
They
Yeah, so like it is the effort of systems biology research is to like taxonomize all the little motifs and organize them into a coherent like taxonomy and find out what biological systems they occur in. So the work of biology is explaining regulatory networks. Like what chemical process produces this regulatory network? So they might try some chemical process with a Petri net, and then think about dynamics for the Petri net. And if you find a chemical reaction that exhibits the right regulatory behavior, then you've like explained that regulatory phenomenon. And biology is really about this. They can only measure like pairwise interactions because they're very limited in the data they can gather. So how do we help them? How do we formalize this process of like, you observe some biological thing and you're trying to explain it with a chemical process? This is where it's really important that we're able to like rapidly change our data model. So we tried to explain this with Petri nets, but it doesn't work. You have to introduce these links in order to get the right answer. And then all of our tools, because they're like kind of parameterized by the schema, like, or any category, there's all these results that are like, for any category with finite products, you can make this structure, right? Because we work at a higher level of abstraction, we can just rapidly change schemas. So we want to set up this inverse problem of like, find me a Petri net that explains this regulatory network. And there's kind of 4 cases you have to think about. The input-output pair transition, input to transition with incident link, output from transition with incident link. And they give you kind of these primitive behaviors. So from these kind of 4 cases, you can come up with a functor from signed Petri nets to signed graphs that interprets a Petri net in terms of what are the observable regulation patterns that this Petri net would cause. And then explaining observed phenomena becomes asking for like a pre-image under this functor. So you say, I have a signed graph, which I measured. What is a Petri net that has these, that would cause these interactions? And any one of them will do, except some are better than others. So you want to figure out, like, given your regulatory network, what's the smallest Petri net that would explain it? Something we haven't worked on yet, but would be necessary to make this really practical. Like, computing things in this pre-image would need to be an algorithm that you could do quickly. I said, why do we need links? If you just try to naively like go from Petri nets to reg nets without links, you'll get the wrong answer. And so you're able to like construct little examples. And the requirement of functoriality, it tells you, like, it gives you a standard by which to judge a construction. If you're just thinking set theoretically, like for every Petri net, I need to get a reg net, and that needs to be a function. Like that's not enough mappings. That's not enough information to be determined, right? Like there's just too many of those functions. It needs to be a functor from the category of Petri nets to the category of signed graphs, or else it's not, like you could just do it any way you want, and it's not enough information. So not only do we have tools, we have kind of aesthetic criteria that you can use to tell if you did a good job. All the constructions have to be functorial. It's like, tells you if you did a good job. So at this point, we can use, this goes back to like 2 years ago, ACT paper we had. We can use regnets as a graphical language, or we can use graphical languages for building systems, and then we want to give them dynamics. So this goes back to like Baez and Fong. You have some combinatorial structure representing the data of the model, and then you want to build a dynamical system functorially that like does the behavior of the model. Yeah. So another key thing that I think makes our approach special, our being ACT, is in traditional CS and math, you have a very small number of like well understood categories: dynamical systems, smooth functions, matrices. Like you have trees, graphs. You have very limited pool of ways of representing information, and you have to embed your domain-specific problem in one of those like 6 options. And so it's like very elaborate encodings that people come up with, right? But in ACT, we'll just like invent a new codomain. Like if we can't find a functor from our domain into the codomain we want, we just change the codomain to have more structure or to capture what you want. So we took like dynam and we went to paradynam. So the objects of paradynam are like the set of state variables, the set of parameter variables, and a parameterized vector field, which is just a function from the like r to the p into dynam on S. Right? So for every point in the parameter space, you get a vector field over the state space. Very natural notion of what it means to be like a parameterized vector. So then what are the morphisms? Well, they're going to be a map between the parameter spaces and a map between the state spaces, so that when you like transform the parameters and you transform the vector fields, this square commutes, right? So it's like a reparameterization of both. It's a reparameterization of both. It's like a change of coordinates for the state space and a change of coordinates for the parameter space, so that the like parameterized vector field coheres. Cool. And like, thanks, general abstract nonsense, for showing that paradynam has finite colimits, in the previous century. So the classic predator-prey model. So like, how do we give dynamics to regnets, right? Like, they, by themselves, they're just like up, down, up, down, up, down, no numbers. Like, how do you make a quantitative model? So Lotka-Volterra dynamics are like the simplest nonlinear equation that people use in practice. And they give these nice little like polynomial vector fields. They're parameterized. They're basically like only involve quadratic terms. And that's nice. And they're like realistic models of ecosystems. So there's the 2-variable classic predator-prey model, and then the arbitrary number of variables kind of linear algebra formulation. Just like replace numbers with matrices and go. So one like critical thing about biological processes is that they're non-negative, because you can't have a negative number of cells. You can't have a negative number of molecules, plants or animals, whatever. So we like redo the whole paradynam thing with conic spaces rather than vector spaces. So you can take convex combinations of the parameters and you can get convex combinations of the state spaces, type stuff. And then you get like non-negative vector fields. Also has finite colimits. So like, how do I take a finite graph and get a parameterized dynamical system? I can use the Lotka-Volterra equation. And so here you're summing over it's like for all of the vertices, do something with all of the edges incident to that vertex, right? So like the API of the C set, you can ask like, for every edge, what is its source and target? For every vertex, you can ask like, what are its pre-images of the source and target maps? So that's all that you use to write this formula. It's like the API of a C set. It's like directly used in this formula. But like, okay, so finite graphs, we got paradynam, but that positivity thing was bothering us. Like, why? Like, where'd the signs go? So if you have a finite signed graph, you can go into paradynam plus, where you're actually like enforcing the signs. So this is really useful in biochemistry, where everything degrades by default. Just molecules like exponentially decay by default, unless you make more of them. And then the signs come in here. Great. So we use a bunch of different categories. These are like section numbers from the paper if you want references there. Everything lives to open systems by just doing structured or decorated cospans as appropriate. And you can build like open sign cat, open sign graph, open paradynam, and then you can build these systems compositionally. So yeah, we have this like robust toolbox of technologies that's ready to go. Abject, slice categories, Kleisli, dynam, structured and decorated cospans. Like you can just pick a science problem, take those tools, and see if they work. And odds are, at least I think they work in general. And yeah, this can be used to push the Pareto frontier of usability, generality, and performance in scientific modeling. We sat down and wrote, skip, don't verify garbage. So like we took this paper and we're like, okay, we need to make a package for doing regnets. And this is all of the code. These includes are like not that important. So this is the most important file. It's 52 lines of serious code. And that's like the whole implementation of most of the ideas in the paper. So and you can like just sit down and write it. So yeah, we're hopefully being able to like have impact in biology. That's the next step. It's like actually talk to biologists and try to use this to solve their actual problems. I'll take questions. Thank you.
Time for 1 or 2 questions, so can see some hands.
What is this? Okay.
Talking to the top.
Oh, cool. You said 1 or 2 questions. I have 2. Shall I just ask 1?
Yeah.
Okay. So you said a load of stuff that sounded kind of logical, and you mentioned functorial data migration, and then you described this sign graph category, and you said, okay, well, it's this slice of graphs, and graphs are the topos, so I think signed graphs are the topos too. And so my question is, have you thought much about talking about these motifs using the internal language?
Yes. We're always trying to make this happen. Yes, so we have like a general implementation of the logic internal to pre-sheaf toposes. And it's fun. The only thing that I've successfully, every time we find a new pre-sheaf topos, we're like, internal logic, it's gonna be useful. And the thing that is always useful is that it lets you define induced subgraph for arbitrary schemas. So you can say like not, you have a subset of the vertices and you just like double negate and that gives you the induced part. And like that's useful. And then like, we're always trying to make it happen.
Thinking more like you mentioned things like if you upregulate this and downregulate that, then this happens. And if you build a system out of components, then that other thing happens. And it sounds like those kinds of things are logical statements, and you might want to express that language. And lift it to the logic of the dynamics in some sense.
So one way of building big motifs would be taking small motifs and combining them with the logic. I think that would be one way to build bigger motifs.
There are more.
But yeah, I don't know how to connect this. I think this is like a huge open question that people should work on, is how to connect the structural information about the models. Like it has this motif. This is, mouse is backwards of my laptop. This one, like what about the structural information of this graph, sign graph, causes this shape in the dynamics curve, right? That is the central question of differential equations in biology. They look at this graph, they make these plots, and they want to know why. I don't have an answer. Grand chat, I think it's the most important question in ACT. Like, well, as Jules calls it, applied category theory. Go ahead.
Hey, can you hear me? Oh,
yeah.
Hey, whoa. I just wanted to use the box. Great presentation. I just wanted to say, I think this is really the perfect blend of theory and practice for the ACT conference. It goes into enough detail that you can see the category theory, but also you're enough into the discipline itself. And I'd like to see more presentations like this vein. I think that's going to be very useful moving forward.
Yeah, and it takes like, because I met Adam McLean at a tumor, like immunotherapy for cancer workshop, like not at a math conference, right? So that's really, we need to take inspiration from the domains. If you want to I can repeat the question if you just shout it.
I'm sorry, I think we have to move
Okay.
on. So let's thank our speaker again, everyone. And James around. This is
Okay.